All right, well, I will go ahead and start. Good afternoon, everyone. I am Marie O'Neill. I am the Director of the Integrated Health Sciences Core at the Michigan Life Stage Environmental Exposures and Disease M-LEEaD Center here at the University of Michigan, and we are funded by the National Institute for Environmental Health Sciences. I'm very pleased to welcome our speaker for today, Ariana Haidari, who has a Master of Sciences and is a registered dietitian. She is a doctoral student here in our Department of Environmental Health Sciences, and she's doing a dual PhD in scientific computing and a certificate in science, technology, and public policy. She's interested in using multi-omic methods to identify preclinical effects of exposures and improve precision in environmental epidemiology. Her research looks at how toxicant exposures across the lifespan alter the epigenome and how these alterations are related to detectable changes in biomarker and metabolomic measurements. A registered dietitian since 2016, Ariana is also interested in relating multi-omic data in clinical practice. She's the founding coordinator of the Weight Inclusive Practice in Healthcare workshop, which trains future clinicians to provide weight-inclusive medical care. Ultimately, she hopes to use her work to improve policy that prevents harmful exposures at the individual and societal scale. So welcome, Ariana. The floor is yours.
Thank you, Marie. I'm just going to take a second to share my screen here. All right.
While you're sharing, I also neglected to mention, as people have questions, please put them in the Q&A, and after Ariana's talk, I will read those to her. Thank you so much.
And can everyone see the slides okay, and only the slides?
Yes.
Great. Awesome. All right. Well, thanks, everyone. So yeah, as I had in the title of the talk, this is going to be a presentation on the effects of gestational phthalate exposure on age-specific DNA methylation. And thanks so much for M-LEEaD for inviting me to be a webinar presenter. Before we get started, I just want to make a statement on generative AI. So I do not use generative AI in any of my work or personal life. And this presentation is a product of my original work, including all text, figures, background research, presentation scripting and storyboarding, and any figures that I use that I did not generate myself, I included their citation. So just that bit of disclosure. All right, so today we will talk about two key topics, toxicant exposures and the epigenome. Specifically today, we'll talk about phthalates, including what they are, how we're exposed to them, and why we should be concerned. We'll focus a little bit of this talk on why we're concerned about gestational exposure specifically, and I'll take us through some of the research I've been doing to better understand how these exposures impact our health. And then we will wrap it up. So to talk a little bit about phthalate exposure, first of all, what are phthalates? So phthalates are plasticizers that are found in lots of materials. They're in personal care products and enhance fragrances. They are added to food packaging. They're found in cosmetics and nail polish. And basically, they're in most materials that are made of plastic and are flexible because they increase the flexibility of these plastics. The problem is that they are not covalently bound to the products they're applied to. And so they leach into skin or into the food that they're surrounding, making them easily available throughout our environment. So when we are considering where we might find these products, we really know that they're everywhere. And they've been found in actually every human sample. And because of this, they've been termed everywhere chemicals. So you might have heard of forever chemicals. Phthalates are a little different. They're more termed the everywhere chemicals. The main concern around phthalates is that they interact with hormone signaling and are therefore termed endocrine disruptors. And we also know that they cross the placenta and have been detected in umbilical cord blood. There's been multiple different studies that have found different health effects related to phthalates, and I'm going to get into those next. When we think, and we specifically want to think here about gestational exposures and phthalates. So they've been related to DNA methylation at birth, where one study found that prenatal phthalate exposure was associated with differing levels of DNA methylation. We also know that prenatal phthalate exposure is associated with gestational age and preterm birth. And other studies have also found that prenatal exposure is associated with delays in childhood. And this is just talking about the prenatal exposure. There's also been a very large body of research looking at phthalates at multiple different time points and how they interact and are associated with health outcomes throughout the lifespan. So if we want to understand how our health is impacted, we need to think a little bit more about how we can design some studies to understand these issues. So I'm going to take a step back now and talk a little bit about how we can ground this question. And thinking specifically about gestation, we can turn to a theory called the developmental origins of health and disease. So this theory is basically helping us frame the sort of idea that exposure to environmental factors during critical periods of life can change our disease susceptibility later in life. And this could be exposures like diet, like chemicals, like stress. And this was first introduced looking really more at nutrition, but over time we've been able to have studies that have measured a wider variety of different exposures. This was first introduced by, and this is known as DOHAD. So this was first introduced by David Barker, in the 90s, but now there's hundreds of publications, and we've seen some sensationalization of this through the common media, as I'm showing with these Time magazine. But basically the piece to really understand here is that these early life and specifically gestational exposures can not only impact our own disease risk much later in life, but also potentially impact risk of future generations. So one of the primary mechanisms we can use to study this phenomenon is DNA methylation, which is an epigenetic regulator of gene expression. So one metaphor that's used commonly when discussing DNA methylation is that DNA is like the piano and epigenetic modifications are the music. So we have a set DNA that we have throughout our lives, but DNA methylation and epigenetic processes dictate which genes are expressed when. And in the case of DNA methylation, it can be in response to some environmental triggers. So for studying this mechanism, we take measurements of the proportion of DNA methylation at specific sites throughout the genome. And understanding whether there's more or less can help us understand later on in DNA expression how this has been impacted. So I'm going to be talking a little bit about CpG sites moving forward in this talk. So we also want to make sure that we're thinking about not just what the exposures are, but when they're happening. So as we mentioned, with the Barker hypothesis with developmental origins of health and disease, we're looking at these critical windows of susceptibility. So gestation being a main one, and considering this molecular mechanism of epigenetic reprogramming. which is especially important during gestation, but also the fact that during gestation there's other sort of organ development that goes on at that time. Another example of a critical window of susceptibility could be puberty, when we see hormonal changes, changes in insulin sensitivity. And so when we're thinking about how exposures, especially an exposure like phthalates, that's an endocrine disruptor, might be impacting our health, we want to think specifically about how when these exposures are happening, such as during gestation or during puberty. Okay, so to think about how DNA methylation and these exposures are tied together with DOHAD, I'm going to talk through this figure a little bit here. So let's say that we are thinking about our normal trajectory throughout our lives. It's normal for our DNA methylation levels to change as we age. You might have heard about epigenetic aging as a measure of how DNA methylation changes at different time points in life. But if there are certain exposures that are modifying how that methylation is happening, we can think about these exposures as being deflected onto the epigenome. So I'm going to try to illustrate this here. Let's see if these animations work. So maybe we have an exposure early in life that changes the methylation pattern to eventually, as we age, end up at a much higher level of methylation than we would normally expect. This could be an example of environmental deflection. We also could see that there's an exposure maybe that doesn't impact us as much, and we just have a normal rate of age-related hypermethylation, maybe not driven by the exposure, just something that happens. We could have other exposures that might lead to a decreased level of methylation. We could also consider this to be environmental deflection. And then if we think about all of these CpG sites, these sites on our genome where DNA methylation takes place, over the course of our life, if exposures are influencing over the course of our life how this DNA methylation is happening, we might see changes in methylation across our entire epigenome. And so the question here is, what happens if phthalate exposure is one of those exposures? How does it change the trajectory of DNA methylation across our life? We might think of lots of different exposures and how they play in here, but I'm going to focus on phthalate exposures to understand this phenomenon of whether the exposure itself is reflected on the epigenome as a person ages. Okay, so this brings us to our research question, which is, does phthalate exposure during pregnancy change the normal trajectory of DNA methylation across our lives? That is, if someone is exposed to phthalates early in gestation, does this modify the level of methylation that they might have either at certain CpG sites or globally throughout their entire throughout their life? And how this methylation is happening, is that pattern impacted by the exposure? So to understand this in a human population, we need to turn to a prospective cohort study, and I'm very lucky to get to work with the Early Life Exposures in Mexico to Environmental Toxicants, or ELEMENT cohort. It's a really special cohort that's been going on for over 30 years. It's a multi-generational birth cohort starting in 1994 and based in Mexico City. It includes over 1,600 mother-child dyads and includes a huge comprehensive catalog of measurements. So we have information about all types of exposures and also all sorts of outcomes from lots of different samples, including blood samples. But the most important piece of this cohort, the most important variable that we have is time, because with these sorts of exposures, and especially considering the developmental origins of health and disease, we need time to see how an exposure at a low level early in life might manifest later on down the line. There we go. Okay, So to answer this question, we can conduct what's called a epigenome-wide association study. In an epigenome-wide association study, we take a blood sample from each participant and measure the amount of DNA methylation at each of the CpG sites that I mentioned earlier. We want to understand how methylation is changing over time, especially in the context of this prenatal phthalate exposure. So we need to take multiple measurements as the person ages. And this sort of longitudinal modeling is relatively rare for DNA methylation and epigenetic studies. So this is kind of what we're lucky to be able to work with the element cohort. To measure the amount of methylation at each site, we use the Illumina EPIC array, which measures 850,000 sites across the epigenome. We quantify the amount of methylation at the different cell types within this blood sample and pre-process the data to determine confounders. Because we're using repeated measures, we can use a linear mixed effects model where the outcome is DNA methylation at each CpG site. So our first version of this model was CpG site, the DNA methylation as the outcome, and then phthalate, age, some principal components of variation related to the measurement, the cell types included in this blood sample, and sex as confounders and covariates. So this first model tells us if phthalate exposure is related to changes in DNA methylation at all. But this doesn't answer our question if phthalate exposure is related to DNA methylation as a person ages. To answer that question, we need to run another model, which includes an interaction term for phthalate and age. And this interaction terms allows us to understand whether at specific ages is the effect of the phthalate significant on the DNA methylation level. So with the element cohort, we have, as I mentioned, multiple time points. So there's a pregnancy time point, which is when the phthalate measurements were taken. Then we also have a delivery time point where there's cord blood that was provided and DNA methylation was measured in this sample. And then there's another sample from childhood, which also allowed us to measure DNA methylation. And then finally, in the teen years, we were able to measure DNA methylation again. The initial analysis, which included just the DNA methylation by itself, included 89 participants that had at least cord blood and one other measure, so childhood or adolescence. But then for the main analysis here, what we really looked at were these 70 participants that had their mom's prenatal or their mom's urinary phthalate measurement, and then the cord blood, and then a childhood or a teen measurement. So to measure phthalate exposure, 3 urine samples were included, and then we took the geometric mean of those samples and adjusted for specific gravity. So you can see here the 5 phthalate species that were included. For DEHP, we summed the DEHP metabolites and included that as a summed exposure measurement. And for the other 4, we just used the metabolites themselves. So for our first model, the one that does not include the interaction term, the one that's just looking at is phthalate exposure, prenatal phthalate exposure associated with DNA methylation across childhood. We found 13 CpG sites where it was significantly associated. And I just showed in this table here the 5 different phthalates, the different CpG sites, and then which genes these CpG sites were on. Not every CpG site goes directly to a gene in this data set. So that's why we have some gaps here. But the more exciting results, as I'm getting to here, is the interaction of gestational phthalate exposure with DNA methylation. So here we see that there were 21 CpG sites that were significantly associated with gestational phthalate exposure as the child ages. and I'll just pause for a second here to show these. To visualize these results a little bit better, though, what I really want to show are these spaghetti plots. So these plots are showing the DNA methylation at the 3 different measurement time points. And to make it a little bit easier to see the separation here, I colored the highest quartile and lowest quartile of exposure. So the participants who had the highest phthalate exposure quartile were in the 75th percentile. We're in the yellow color, and the ones in the lowest percentile were in the blue color. And what you can see here, and again, there were 21 sites that were significant for this, but I'm going to show you a few that are just the most easy to see the divergence. What you can see here is that the separation between the highest exposure quartile and the lowest exposure quartile really starts to expand as the child gets older. So we can see that in this case, this is MBZP. There is a different level of DNA methylation depending on exposure quartile. So here's another figure. This is showing the interaction effect of MEP in age on DNA methylation. So again, you can see that the higher, in this case, actually, I should say it's flopped. So the 75th percentile is actually in the less methylated kind of vague section here, and the 25th percentile is in the higher methylation level here. I should add that in general, this is probably not related to any major difference in expression because, generally they're in the same amount of DNA methylation, but we can see that there is a little bit of separation, and we want to understand whether or not that was important. So more on that in a moment. And one more plot just to visualize this relationship a little bit more. This is the interaction effect of MCPP in age on DNA methylation. So we see that there's in the 25th percentile category is kind of all clustered around here. And then the higher exposure level is separating more towards the top. Okay, so to better understand these DNA methylation changes, and whether they could be related to actual changes in health, we completed a gene ontology and KEGG pathway analysis. And so we do this to understand which biological processes and functional pathways might be affected based on which CpG sites were affected by the exposure. So we used the R package mismethyl. And to do this, we included the top 5,000 CpG sites for each phthalate exposure model in the epigenome-wide association study. So we took, for each of these models, we had run 850,000 models, give or take, after cleaning. And then we took all of the results and chose the top 5,000 for each phthalate and included them in these pathway analyses. And then we do FDR correction to make sure that we're not, so that we're more as conservative as possible with the significance of these pathway analyses. And unfortunately, after doing so, we didn't have any significant pathways, but we were able to get several hits that came close. They're p-value of 0.01. And in the GO pathways, interestingly, synaptic signaling, membrane structure, and some other neuro-related pathways were implicated. And in the KEGG pathways, which measure function, we saw neuro and cardiometabolic pathways. So this is definitely interesting because we know that phthalates have been associated with neurological outcomes in other studies. And if we're really considering what we're looking at here, we're talking about an exposure that's very small, that's during pregnancy, before the child is even born, and how this could be impacting them, 15 years later in some cases with the time delay between the first, second, and third DNA methylation measurements. So our next step here is to measure whether this is actually leading to a change in RNA sequencing, or sorry, in DNA expression using RNA sequencing. So we want to look at how these sites that have differential methylation based on phthalate exposure at specific age time points could be potentially expressed differently. And that would really tell us that an early life exposure measured, again, before the child was even born, could be impacting health outcomes much later on in life. Okay, so what else will we be doing next? So another kind of key question here is how this could be actually, impacting some of the sort of other disease processes that we know are related with the outcomes that we're thinking about. So the next thing we want to look at is how maternal diet, the relationship between maternal diet and DNA methylation could be mediated by gestational phthalate exposure. And the reason this is important is because a lot of phthalates are, one major route of exposure is through processed foods. And of course, there's a lot of concern around processed foods and their relation to health outcomes. But when we're considering this relationship, we often don't parse out the contribution that toxicants make to the disease endpoints. So if we include a mediation analysis where we look at the effect of the phthalate exposure, we can understand, is it the food itself, or is it, and then the nutrient components of that food potentially, or is there a component of this exposure or of this relationship that's actually due to the toxicant exposure that comes along with the food? This is also really important when we're considering sort of the regulatory implications and what direction we want to go with our public health interventions. So what we typically talk about, right, is that a healthy diet is important for XYZ reason, but we might over contribute the impacts to maybe the nutrient profile, but without considering the actual toxicant exposures that come along or don't come along with particular foods. So that's why we want to include this mediation analysis to start to understand how these toxicants are playing into the relationship between diet and, in this case, DNA methylation, but eventually to understand the relationship between diet and chronic disease or any disease endpoint. So in this study, we used repeated measures of the epigenome across childhood, and we found that prenatal toxicant exposure could affect age-specific DNA methylation patterns in childhood. This example is just with phthalates. We want to also understand whether this could be happening with other exposures, potentially exposures that we know have a stronger effect, and understanding where in this DNA methylation trajectory there could be effects of these exposures. So this can help us understand how exposures early in life during pregnancy can impact health across the lifespan. The element cohort is a very comprehensive effort. I wanted to say thank you to everyone who is involved with the element cohort, and also acknowledge my lab. I'm advised by Dr. Jackie Goodrich and Dr. Dana Dolinoy. Also, thank you to Dr. Karen Peterson, Dr. Jenka Wu, and Dr. Wei Peng. And the funding that I've been lucky enough to be provided is from the NIEHS Environmental Toxicology and Epidemiology Training Program, the Global Health Early Dissertation Award, which allowed me to visit Mexico and get to meet our fieldwork team, which was an invaluable experience to actually meet some of the participants and see how this really amazing cohort has been carried out over the last 30 plus years. Also the Career Training Grant in Reproductive Biology and a Ruth Christian National Service Individual Predoctoral Fellowship. Thank you.
Thank you so much, Ariana, for that wonderful talk. Really interesting results, and I will go ahead and start with some questions that have already been contributed by our attendees. So Dr. Devin Payne-Sturgis had two questions arising, the first being whether the DNA methylation that you've seen, has that been associated with specific health outcomes among the kids?
Thanks, Dr. Payne-Sturgis. So I haven't had the chance yet to look at whether these specific genes that we found in this study were associated with specific health outcomes. I know others have looked at some of the other health outcomes in general in the cohort. One of the pieces that I'm really interested in, as phthalates being an endocrine disrupting chemical, I'm interested in whether there's a relationship between these exposures and potential diabetes development, early type 2 diabetes development. So we know that we did look at phthalate exposure early in life and impaired fasting blood glucose in the teens, and did find some results there. So that's kind of the next piece that I'm looking at, is to try to see if there's any outcomes related to that.
Great, thank you. And this is a related follow-on question, again, from Devin, is whether you accounted for continued phthalate exposures among the children who obviously are exposed postnatally, or whether that's captured by the age term in the model.
So yes, I am frequently asked about phthalate exposures in the children and why we don't include those. So when we were talking about how to kind of making our DAG and thinking about the exposures to include in these models, we didn't want to include phthalates twice to sort of double measure the effect of phthalates, because we assume that the household has generally similar phthalate exposure, both from a mom during pregnancy and then her kids growing up. They probably have the same exposure profile. We probably should, and I think plan to do some sort of sensitivity analysis just to see how much of the total effect might be included in the lifetime phthalate exposure. But what we're really trying to capture here is the total effect of prenatal phthalate and DNA methylation. And if we include these sort of intermediate exposures, then we'll be only capturing partial effects instead of the total effect there. So that's our plan.
Thank you. Another question that may require you to refer to the slides again from Yue Zhou, which is great presentation. And for the equation, how did you choose the principal components of variation?
Yeah, this is a great question. I have a slide. I'm not sure if I want to do the whole thing. I'm trying to reshare. Maybe I shouldn't have stopped sharing. But basically, when we do this, when we do the epigenome-wide association study, we look at some of the confounders by doing single value decomposition. And so that helps us, and a PCA plot to help us understand which components of this, of the entire epigenome-wide association study process before we get to the modeling actually could be impacting our outcome. So yeah, these are components that are related to that, not related to like specific features of the participants or anything like that.
Great, thank you. Haley Jenkins has two questions. One is, were phthalates also measured in 2 childhood urine samples? I know you addressed to some degree the issue of childhood urine samples, but she continues that it would be interesting to incorporate that into the project to see how estimated cumulative exposure could impact DNA methylation, especially looking at high gestational plus high childhood exposure versus low gestational and low childhood exposure. So maybe you want to address that one, and then I'll share the second question.
Yeah, that's a great idea. Yeah, I mean, I do think that we should also look at the childhood exposures in general. Whether that would impact the DNA methylation, I'm really not sure. I think what we're kind of operating here on is that these changes to DNA methylation are, the pregnancy time point is important for these methylation changes to take place. But you're right, if we do the same sort of analysis, including the childhood exposures, we could really nail that down of whether this is related to the pregnancy time point or if maybe it's something that could be related to childhood exposures.
Yeah, and just to add, I think it would be interesting as well to see if in the families, that both the parent and the child are systematically being exposed to more phthalate exposures, which could suggest potential interventions for them to reduce that possibly.
Mm-hmm. Right.
All right. So Haley had a second part, which is, do you know if there is any history of phthalate regulation in Mexico? If so, and if it happened between gestational and childhood measures, so the time point, it would be interesting to see if those had an impact on exposure and in turn DNA methylation.
Yeah, that's a great point. I know that in general, DHP is, has been phased out. I would have to check on the dates in terms of Mexico. So I'd have to get back to you on that. I should know that. But yes, we know that the pregnancy time point is more likely that there's more exposure as opposed to what's happening currently now or even during childhood. But the childhood measures themselves are still as recent as the latest teen measures are, I think, 2015. So yeah, I'd have to double check on the time point for that.
Okay, thank you. Another question, Justin Thompson wanted you to please clarify the 13 CPG sites and the 21 CPG sites that were significant, and wonders if those overlapped or were they completely distinct sets.
Sure. Yeah, I didn't include the tables that I have of those CPG sites because they're mostly just a lot of numbers. So they do not overlap, which we thought was really interesting. I could reshare the slide with the genes if that would be helpful. I'm happy to pull that back up. Maybe if, Marie, if you want, I don't know if you want to go to the next question, but I can pull that.
Oh, sure. Yeah, while you're in the process of pulling that up, it seems that there's one question in the chat. Could you please explain how you use KEGG in your analysis? And is it possible to incorporate effect size, direction, positive or negative, and p-values when using KEGG? Thank you. I know you wanted first to show the 13 CPG, so I'm happy to repeat that if you...
No, that's fine. I guess what I'll say is I don't have the table of those CPG sites in this presentation. I'm happy to follow up with the question asker regarding those specifically, but here I'm just showing that these are the different genes that we were able to find that those CPG sites are on. So here is the first model that just looks at phthalate exposure, and this is the interaction model that uses phthalate by age. So yeah, unless you have CPG sites memorized, I didn't want to include those in this presentation. I don't even have CPG sites memorized, so yeah. But thank you for the question. And then the next question being about the KEGG pathway analysis.
Yes, and whether, incorporating effect size, direction, and p-values when using that method.
Yes. So I would have to get back to you talking about the effect size and direction, but this is the list of KEGG terms, which I didn't include in the presentation again because just trying to have it be a little more interpretable in terms of the discussion. But these were the KEGG terms that had a p-value less than 0.01. So as I mentioned, none of them were significant after FGR correction, but we did have several that had a p-value less than 0.01. And you'll see that there were a few that showed up all related to neuro pathways came up in the presence of multiple phthalates. So the way to read this is what's the name of the KEGG pathway, and then which model did it come up in. So we have a different model for each of these 4 phthalates. And there were none for DHP, so that's why there's no column for DHP.
Thanks. Yeah. And so the FDR is the false discovery rate. So it's a way of accounting for the multiple comparisons. Good to know that you did that. Okay, great. Another question is, why do you speculate that MEP had a reverse relationship with age compared to the other phthalates?
That is a great question that I do not have a good answer for. I'm very sorry, Karen, but yeah, I don't know why MEP specifically would have the reverse relationship. I do, let me see if I have one here. The kind of thought that I've sort of been going back to on some of these is that we've had a lot of sort of surprising outcomes in terms of we have an environmental exposure and we think it's going to, increase inflammation, but it decreases inflammation. Thinking about, are PFAS actually good for us, or phthalates actually good for us? Of course they're not. So yeah, I definitely need to think about this a little bit more about why it would have a different outcome compared to the others. Yeah, I'll have to dig into that a bit more.
Great. Here's another question that overlaps with one I would have, which is, are other toxicant exposures collected, and how correlated are phthalates with these other toxicants? Leading to the mixtures question that I'm sure you've also thought about.
Sure. Yes, other toxicant information is collected. That's one of the amazing things about Element. There are lots of other exposures, including heavy metals and other endocrine disrupting chemicals. But in terms of how correlated they are, I haven't looked at them in particular with this specific data set of these 79 people. I haven't done correlation of the other exposures on this. Do you expect the other endocrine disrupting chemicals, some of the phenols, for example, to be correlated? In terms of mixtures, the reason we haven't done mixtures for this analysis, at least initially, is because we have these 850,000 models, doesn't really translate very well to the sort of mixture methods. Although what we're thinking we'll do next, instead of using it for the entire epigenome-wide association study, is now that we have a very specific list of genes here, we can do a mixture analysis on, at least on the phthalates, but like the phthalate mixture for these very specific genes or very specific CPG sites. So stay tuned for that.
Great, thank you. Another question from Farah Nassar is, did you try to do DMR analysis?
I have not. I have not done DMR.
Okay. And I think we have gotten to all of the questions. I would like to ask a somewhat practical question, Ariana, which is, how can people decrease their phthalate exposure?
Yeah, so I think this comes up a lot in public health discussions, is how we can decrease our exposures. I think there's a few simple things that come up a lot in discussion around not microwaving plastic, not heating plastic, as much as possible, the vegetable bags that you can microwave them in the bag, sort of avoiding that sort of thing. But I also really think the key piece that we need to be discussing more as public health, both scientists and myself as a clinician, is really talking about how this is a policy level issue. This is something that we really need to see federal policy that reduces our exposure and actually tracks and bans and decreases the sorts of chemicals that we can be exposed to, especially through our food and in our day-to-day product usage. I'm pretty hesitant to go down a path of telling people to avoid this and avoid that specific thing, because I think a lot of the time the burden for that falls, on whoever is like the caretaker of the home, kind of contributes to guilt and anxiety and all these sorts of things. And the other side of that is that we don't know, if you cut out this one specific plastic or these two specific plastics from your life, is it going to lead to a meaningful difference in your health outcomes, which, of course, we want to be aware of those things. But I think that it's very important that we're clear about what these changes actually, what the payoff of these changes actually is, because we have to balance sort of the societal reality of these exposures being everywhere. So what does that mean, in comparison to the types of changes that we can make? That's not to say that, we shouldn't be aware of products that have a lot of phthalates, that plastics that we really don't need to be using in our lives, that, things that we're heating, again, as I mentioned, putting the hot leftovers right into the plastic container. We probably want to get away from that if possible. But I always want to balance that comment with really thinking about like what is the main goal here, which is better, stronger policy that protects people in all walks of life at all stages and isn't dependent on an individual's knowledge, energy, access. And is really protecting everyone. So yeah, those are my thoughts on that. But
yes, thank you very much. I think that was really important comment, and especially I can see that you're doing very rigorous research, asking a specific question about an exposure that is ubiquitous, but keeping in mind the big picture public health implications and wanting to prevent exposures that may be damaging to our health. So thank you so much, Ariana, and I want to thank all of those who attended today. Great set of questions. Meredith has kindly put in the chat the M-LEEaD YouTube channel where you can see Ariana's talk, among others that have been recorded, of cutting-edge environmental health sciences research. So with that, I'll thank everyone for attending, and we look forward to seeing you at a future M-LEEaD event. Thank you so much.
thank you.